This week I'm with Lynne Boddy, MBE. Lynne is a Professor of Fungal Ecology at Cardiff University UK. Her research focuses on how fungal mycelia behave in the natural world, climate change effects on fungi, antagonistic interactions between fungi wars, and development of fungal communities in wood. Welcome to this week's edition of Tree Lady Talks. She is currently studying the fascinating communities of fungi and other organisms that rot the centres of old tree. She is a prolific author, having co-authored Fungal Decomposition of Wood and The Fungi, and her most recent being Fungi and Trees, Their Complex Relationships. She also has written a children's book, Humongous Fungus. She was a president of the British Mycological Society. She's an ardent communicator, as you'll hear, on the mysteries and importance of the amazing hidden world of fungi to the general public, including TV, radio, popular talks, videos, articles and exhibitions. She was awarded an MBE in the Queen's Birthday Honours List in 2019 for services to Mycology and Science Outreach. So welcome to Professor Lynne Boddy from Cardiff University. Lynne, many of us know you already, but I always find it fascinating to hear about how you began. So tell us about your early career, please.
Right. So I guess you want to know how I became interested in fungi, really. Well, it was as an undergraduate at the University of Exeter. And many people think, oh, well, that must have been because of the very famous mycologist there, Professor John Webster. And indeed, he did teach me, which was splendid. But the thing which really intrigued me about fungi is when we lived in a shared house. I hasten to add that it wasn't owned by the University of Exeter. It was somewhere in Exeter. We lived in the basement. It was a Georgian terrace. What wonderful terrace of fantastic Georgian houses, except for our one on the end. And one day I tried to open some drawers, and they wouldn't open. And we tugged and tugged on these drawers, and eventually they did open. And the reason they'd been welded effectively shut was because mycelium was growing actually through the wall of the house into the chest of drawers and into the drawer itself. And this was my first encounter with the dry rot fungus, Serpula lacrymans. And of course, we happened to open a cupboard under the stairs, and there were these huge fruit bodies making these rusty brown spores and masses of mycelium. And, you know, that's enough to inspire anybody, I think, to be...
Oh, God, we'd all love that. Yeah.
Absolutely. So that was my first great encounter with the power, if you like, of fungi. And I was an undergraduate, as I said, at Exeter, and I did a degree in biology and mathematical statistics, and all of the biology was ecology. And I was interested in any and every sort of ecology, so, you know, marine ecology, freshwater ecology, plant ecology, soil ecology, anything. And I think I would have been very, very happy being an ecologist of any type. But when I graduated, there was a position advertised at Queen Mary College, London, to look at the ecology of wood decomposition on the forest floor. And I was lucky enough to get that job. And the rest, as they say, is history.
And at that time, was it quite a strange subject to study? I mean, what were the attitudes towards fungi and trees and fungi at that time?
Yes, I think it's very different probably from now. Of course, we knew very much less then than we know now, inevitably. So in many ways, we were starting at the beginning because the work that I was doing was looking at how much dead wood there was on the forest floor. So something as basic as that, we didn't really have very much information. There'd been work done in North America, but we didn't really have anything done in Britain. So we didn't know much about it. And also, I think that perhaps people had a bit of a strange feeling, I suppose, about fungal ecologists. And I think that they sort of rather derogatorily thought of them as people walking around the woodlands with a collecting basket and picking mushrooms and doing not much more than that. And actually, I think going around the woodlands and looking at fungi is very important. I think we've come a full circle now to people realizing how important scientific natural history is. But I think that perhaps at the time, I mean, I may be wrong, but I think people sort of look down on that rather. And so I didn't want to be, I was just a young student starting out. I didn't want to be looked down on, right at the very beginning. And so I tried very hard not to be considered in that sort of way. And I suppose in some ways, I didn't go down the pathway of trying to identify fungi here, there, and everywhere, and trying to focus in on various aspects, such as wood decay, and to do ecology in the laboratory as well as in the field, and not to be considered just as an identifier or something like that.
Yeah. Yeah, to look at what and how rather than the name.
Exactly.
And and really fungi are the building blocks for everything, aren't they? Because they are on the earth, 700 million years, way before plants. And where would we be, if at all, without fungi? I mean, what do they do for us?
Well, absolutely. It really was fungi which allowed plants to colonize land, I don't know, 450, 500 million years ago, or whenever it was, because, of course, life evolved in water, and early plant life was in water and sort of bathed in a nutrient solution so they could get their food and their water requirements just from everything that was around them. But when they started to colonize land, it was a very different situation because, of course, it's very much drier on land. And of course, they didn't have roots then. So how are they going to get mineral nutrients? Well, fungi were there. Fungi sort of had the opposite problem with their fine filaments. They could get nutrients because they could bore into any dead stuff there happened to be around, but also into rock and the like, and could get water from all sorts of places. And so it's sort of, if you like, a match made in heaven when plants and fungi got together, because the fungi couldn't make their own carbons and energy sources, but plants can. And plants have the opposite problem. They couldn't get their water and mineral nutrients easily. And so this relationship between plants and fungi has carried on ever since, with 90% of plants having mycorrhizal associations, which is the association of fungi with the fine absorptive roots of plants. And they are this great partnership which really powers our terrestrial ecosystems.
And as well as that, I mean, without the decomposition by fungi, we would be overwhelmed, wouldn't we? We'd have so much organic matter that I'm guessing we wouldn't function.
Yes, that's spot on. There are two main reasons why we wouldn't be here. One is these mycorrhizal associations, and the second is fungi are the great decomposers. There are obviously other decomposers, many of which play a role in all sorts of types of decomposition, but it really is only fungi, and then a relatively narrow group of fungi, which have the ability to break down lignin at a reasonable rate. And of course, as we know, much plant biomass, we think obviously especially of trunks, woody trunks, are made up with lignin or lignocellulose. And so that has to be recycled, because to keep systems in balance, you just can't keep having dead stuff building up. We'd be up to our armpits in it otherwise. And more importantly, nutrients would be completely locked up in that dead stuff and would not be being made available again to plants. So the decomposer fungi break down the dead stuff, releasing the mineral nutrients locked up in there for plants to use again to carry on growing, and the system goes around and around, and here we are very many millions of years later.
And people think of fungi as a pretty toadstool on the ground, but really that's just a small part of it, isn't it? What's going on within the substance that the fungi is fruiting on?
Yes, fruit bodies are just the tip of the iceberg. The main part of the fungus is, of course, the mycelium. For most fungi, for some fungi, it's a yeast phase, but for most fungi, and especially and including wood decay fungi and the mycorrhizal fungi, it is these fine filaments, this mycelium, this network of fine filaments called hyphae, which is the main body of fungi in soil, in wood, in dead stuff, in very many places. And because hyphae are very fine, they have a large surface to volume ratio for soaking up water and nutrients, because they form networks. They have all of the beneficial properties that networks have. So if part of a network is damaged, there are other ways that nutrients and things can flow around these mycelial systems to get to the same place, for example. So it is the mycelium which is the key to the lifestyle of most fungi.
And so this mycelium, so it is an adaptive network that's dynamically changing, presumably due to its changing environment. I mean, what is the optimum soil conditions? Let's talk about mycelium in soil. What sort of conditions do fungi need to thrive underground?
Yeah, that's a good question. I think mycelia can actually thrive in all sorts of different environments. And another benefit of being a mycelial organism is that different parts of that mycelium can be experienced in different conditions at the same time. But because you can have movement through these mycelial systems, one part of a mycelium, say, which is in a patch of lots of nutrients, might be able to help another part of the mycelium, which is not in a place where there are a lot of nutrients, because things can be moved around these systems. So answering your question isn't really straightforward, because mycelium sort of integrates its activities over spatially distant areas and also over time. But of course, if we're thinking, if the question really is the question of how do we, what conditions do we want our soil to be in to benefit the mycorrhizal partners of, say, trees? And that's a slightly different question, I think. Obviously, the ideal conditions would be a reasonable amount of water and mineral nutrients, but not too much in the way of nutrients. So as humans, a very bad thing, which we're doing all the time, is polluting our environment. And that includes polluting with nitrogen. So nitrogen is a big problem. And too much nitrogen in soils is not a good thing, because the mycorrhizal partnerships that you would normally find are lost, and the tree may well make mycorrhizal partnerships with other fungi, but perhaps which are not so good at acquiring, say, other nutrients. I think that's a rather long-winded way of saying, really, that we don't want to pollute, because we're really changing the balance of the tree mycorrhizal relationships and the partners that they have, which is essential for their growth. So we need reasonable water, a reasonable amount of nutrients. Also, it's important that the soil is well aerated and not compacted. So all of these things are essential both to the fungi and hence to the trees as well.
In recent years, there's been a huge interest in the concept of the Wood Wide Web. I've noticed that people I speak to, and you see in the media, people very excited about the idea of there being chattering underground, of this communication of the mycorrhizae network, and a sort of altruistic fungi system to help trees in need, mother trees, saplings, and a sharing of resources where, let's say, there's a fir tree in the winter with excess that feeds a birch tree as it's emerging in leaf, and in the summer that birch tree with an excess of sugars or chooses to share with the fir tree in shade. Is that oversimplistic? Because the particular things that are talked about as being shared are nutrients and carbohydrate, and chemical messages and electro messages. What are your initial thoughts? And perhaps we could break that down.
Okay, so we know that mycorrhizal fungi are essential to trees. We know that both mycorrhizal fungi and decomposer fungi form networks. They form networks on the forest floor. And I've always loved the idea of calling this the Wood Wide Web, because I think that this is something which really brings people in, this idea of this underground network. And yes, mycelia absolutely do move nutrients around from one place to another. We've demonstrated that ourselves with decomposer fungi on the forest floor, that you can put, say, phosphorus in one place and you'll detect it very quickly, several meters away in the mycelial system. Of course, the same sort of thing is true with the mycorrhizal fungi, and lots of work has been done in the lab. In the laboratory, experiments have been done with decomposer fungi, the fungi that I work on, showing that a piece of wood decomposing in a certain place will move nutrients to a piece of wood that is colonizing somewhere else. And likewise with the mycorrhizal fungi, experiments have been done showing that in these little microcosms, that a tree, or I suppose really we shouldn't call it a tree when it's going to be very tiny, probably only about 20 centimeters tall or something, a seedling or perhaps a sapling, if you label it with carbon-14 dioxide, the leaves will photosynthesize, they will capture that radio label, and it will then label the sugars that they make, and those can then be detected in the network underground, and indeed can sometimes be detected in a neighbouring seedling or sapling or whatever. So on a small scale, those sorts of things have been done, both for the sugars moving from one tree to another, and indeed for nitrogen and phosphorus and water. And also, as you've suggested, chemical messages moving through these systems. That's been done on a small scale in the lab, or in perhaps sometimes small experiments outside. And that's undoubtedly true. But then we've taken a great leap of faith in thinking that this is happening on a large scale in a forest from one tree to another, and maybe it does. But I'm not sure that the evidence is there yet. And then, so I'm not saying it doesn't happen, but I'm saying that we need to do a lot more science to test these hypotheses that nutrients might move from one tree to another. And then it's always, as humans, we always try to put things in an anthropocentric or anthropogenic, whatever sort of way. And I think that the mother tree idea is something which we rather like, because we like to think of nurturing less well-off or younger things. But I'm not sure that the evidence really is there yet for that.
Oh, area for more research.
Absolutely. And I think lots more research needs to be done in this direction to understand what really is happening and why, without putting our human sort of connotations onto it. I think what we really need to do is to do a lot more research and set up hypotheses which we can test actually in the field, not just in smaller scale in the laboratory.
I think that's really important, because you're so right. Everybody loves a story, and it's a story that reaches people's hearts and makes a change. And that story is, there is an intelligence underground that is kind in sharing. Now, I really hope that is true. But what you're saying is why I'm speaking to you as a scientist, is that we don't yet know. Let's do some proper research in the field. So, yeah, thank you for that. That's really, really interesting. But we talk about this connection, and indeed, I've been using the phrase on every podcast, trees are holobionts. Indeed, we all are. Every living thing is a holobiont. It's not its own entity on its own. And perhaps you could describe to the listeners, from seed germination through to advanced veteran stage, the role of fungi, just very generally, in the start and the eventual decomposition of that tree.
Yes. So in recent years, I think we've really become aware, both as humans and for other organisms, as you say, that that we don't exist on our own. We know that our own human bodies are coated both outside and to some extent inside too, for example, in our guts, with microbes, and without them, we would not exist. And the same really is true for trees and other plants, but I think this hasn't really been grasped until relatively recently, at least not by a wide audience. And it's not just at one point in time either that that plants are acting as a holobiome with many organisms helping them to exist, and indeed existing on their own. I'm not putting any connotations there as to why an organism interacts with another. I think that would be wrong to do. It's not just at one time in their life, as you suggest, it's throughout their lives. So if we were thinking about, say, an acorn in a woodland germinating, and as it puts down its little radicle, very, very quickly it would hook into the network, the underground network, mycorrhizal network, and we'll get mycorrhizal partnerships forming very quickly because they are there as the network underground. And straight away, water and nutrients will be beginning to pass to the seedling, and at some point, sugars and things will be passing back when the seedling reaches a sufficient stage to photosynthesize sufficiently to pass on nutrients. But it's not only mycorrhizal fungi, of course, that we're thinking about. I think we're well familiar with that partnership now, but it's throughout and overall over an entire plant or tree that we have fungi and other microbes. So if we start, say, at the top of the tree, on the outside of leaves, there are epiphytic microbes, perhaps feeding just on compounds that are exuded through the leaves. There are also endophytes in the leaves. So endophyte literally means within a plant, and this includes bacteria and fungi. They are in the leaves, but they're not just in the leaves. They're in the shoots and in the roots. They are everywhere. And again, we don't know what half of these endophytes are doing. So endophytes, by definition, are symptomless. So when a leaf has endophytes within it, we as human observers on the outside can't see anything wrong with the leaf or better with the leaf. They're symptomless. But we know that they include organisms with all sorts of different roles. For example, some of them might be mutualists. They might be making compounds which benefit the plant in return for nutrients, say, from the plant. And we know that there certainly are some mutualists. There are organisms, there are fungi, for example, in grasses and other leaves, which produce chemicals which deter grazing by invertebrates, which could be seen as being beneficial to the plant. There are other endophytes which are perhaps parasites or pathogens in waiting, just waiting for that opportunity to actually kill or feed upon the plant. There are also decomposers, saprotrophs, in waiting for the opportunity to be there and to rot the leaf or the branch or whatever it is when it dies. So to be the first one there, like being the first one at the buffet table, eating, you've got the chance to get everything before other things come along and perhaps supplant you. So the endophytes are really important within the plants, but we know relatively little still about their actual roles, and there's very much more to find there. I'm interested in decomposer fungi, so perhaps I can take this opportunity to
Yes.
give you
Yeah.
examples of endophytes in wood decay. So I suppose probably most of the work has been done on endophytes in perhaps in leaves, but they're also important in wood decay. So in trees, as we know, the outside of trunks, for example, is the main water conducting tissue, conducting water from the roots to the shoots and indeed branches. There's the wood. It does the same sort of role, conducts water. And actually, it was one of the first pieces of work that I did when I was a postdoc in Bath and started to look at the fungal communities which were developing in attached oak branches. And what we found was that you seem to get these very long individual mycelia or decay columns within branches. And it was very difficult to imagine that they had developed in the very short time that they seem to have developed in, sort of sometimes less than one growing season. And we suggested that perhaps this was because those fungi were already there within the wood, perhaps just microscopically not doing anything, just biding their time. We didn't call them endophytes at the time because there was a conference that I went to where people interested in endophytes were talking. And we decided that perhaps as the xylem that conducts water is largely non-living, we perhaps shouldn't call them endophytes because we're going to reserve that term for living tissues in leaves and what have you. So I just called them latent propagules, the fungal spores or yeast or whatever they are in the wood. I called them latent propagules, but we can think of them as endophytes. And perhaps if I'd have called them endophytes, my papers would have been cited much more. But there you go. Anyways, so we hypothesized that within the xylem, there was lots of these propagules dotted about here and there. And then if for some reason this part of the tree, the xylem, stopped conducting water, maybe a root was damaged, maybe the crown was damaged and was broken up, but it stopped conducting water and it became aerated, that then those propagules could develop. And if they were genetically identical, when they started to grow out as mycelium, these little bits of mycelium met another identical mycelium of itself, a clone, if you like, they would fuse and give the impression of great long individual very rapidly. Now, this was a long time ago. This was in the early 1980s. And of course, modern molecular techniques hadn't been invented. So we couldn't do things that we can do now to detect these endophytes or these latent propagules. So we did an experiment where we chopped down actually healthy branches of beech trees, chopped them into sections, I don't know, I can't remember, about 20 centimeters long maybe, covered the ends with polythene and cut different areas out so they could dry at different rates. Some of them we didn't let them dry at all. And then we found that these what had been fully functional branches actually had fungus growing throughout. We had the decay forming just like you would see in naturally early stages of decay in beech branches. And we had to do lots of things to convince the scientific public that that these weren't contaminants that we were getting there, but we did convince people. And then people realized, of course, that these things were actually there. And probably at the time, I think that people were still largely thinking that the insides of trees were sterile. So that came as something as a surprise.
Wow. Wow, that's really fascinating. The mind, that the understanding of science at that time was so wrong. When you consider how long plants have had an association with fungi, which I understand is 460 million years. And one of these fungi that springs to my mind that I've seen a real upsurge in is Cryptostroma corticale in sycamore. Is that an example of what you're talking about with a fungus that's within the tree that's triggered by, we've had a lot of hot, dry summers, not this summer, and I've seen a lot of sycamore actually dying from that.
In these very dry summers on beech, you often get these strip cankers, so these large bark, which are dead and they often look black on the surface. And sometimes they will spiral up for many meters up beech trees, and you particularly get these chalky soils, and it is usually following drought. And we first noticed these strip cankers in 1977, 1978, 1990, and those followed drought years. And actually, the fungi, it's not lightning, which is making these great big black lines going all up the tree. It is actually, as you would guess, fungi. It's fungal fruit bodies that you're looking at. And commonly on beech, it's Biscogniauxia nummularia and Utipa spinosa. And we often get cankers on trees, and lots of cankers on trees are formed by fungi on the outside growing inwards and killing. But what these cankers, how these cankers are forming is, it is these latently present endophytic fungi within the trunk of the beech trees, which actually are growing out, and the cankers this time are sort of effectively growing outwards. So what's happening is that in a drought, maybe a root has become damaged again, and hence the tree is less, it's even harder to get water than it would be. These huge volumes of xylem dry out, and those two fungi, which I've already mentioned, actually start to develop, and they sort of come outwards, and they cause a canker on the outside of the tree where they fruit. In the case of Utipa, it's a very dark black colour, which gives the idea of these lightning strikes and will spiral for many meters up a tree.
And actually, because tree species have different characteristics of wood, fibres, and structure, the same fungi can have a different impact on a different species. So the one that comes to my mind is Inonotus hispidus on ash compared to London plane. Perhaps you could just explain why there is a different response. This episode is sponsored by Tree Law.
Tree Lady Talk.
Hi everybody, I'm Sarah, the Director of Tree Law. I call myself the tree hugging lawyer, and I specialise in all things around the law and trees. So if you've got a tree dispute or a tree query, then check out my website and get in touch. I'm at www.treelaw.co.uk.
And so something which I didn't say when we were talking about latency was that subsequently we've actually started to look for latently present endophytes within sapwood using molecular approaches. We find, for example, that Daldinia is found in loads of tree species, and yet what do we normally find Daldinia only on? We only find it usually on ash or sometimes beech, Britain,
Yeah.
or birch in more northern.
Okay.
And so I think that is and in also in ash we find lots of xylaria, acacia fungi. It's because you're right. It's because the wood is different.
Okay.
Don't know exactly what it is. We know that ash, for example, is a very dry wood. We know that all woods have different structures, and presumably it is that which allows different fungi to develop. And I think the communities will be changing with climate change because of different water relations and all sorts of things. But I don't know the answer to this cellular structure, and I think that's a very interesting thing. We need to look more at the cellular level to try and work out what's going on.
I remember meeting you a few years ago on a Thinking Arb Day at Windsor, and we were there with Emma Gilmartin as well, and you had these slices of wood, and it was beech actually, and you really captured my imagination, well so did Emma, on fungal wars, almost like robot wars, and these demarcation lines in the wood. Could you explain what's going on there, please? And what these pseudo-sclerotal plates actually are and mean?
I'd absolutely love to. This is something that has fascinated me for, 40 years, I suppose, getting on for. Fungi are great fighters. We've talked about them so far in mutualistic relationships, but wood decay fungi are certainly great fighters. And actually, this was something really that was looked at even before me by Alan Rayner at Bath University. He first started to look at interactions, and then I carried on for a very long time doing this subsequently. In any piece of wood, lots of fungi will arrive. They might develop from these latent propagules, and subsequently they will arrive as spores, or if it's wood on the forest floor, they might arrive as mycelium, as mycelial cores, something like that. And for these decomposer fungi, they have to get hold of territory in the sense of volumes of wood, because that is the food that they are going to eat. And if they can occupy a volume of wood and protect that volume of wood, then effectively they can decay that wood at their own rate, the rate that suits them. So some fungi actually decay wood very slowly. So lots of the xylariaceous fungi, things like Deldinia and Xylaria hypoxon, for example, they decay wood quite slowly. Other things decay wood much more quickly. Some fungi get in early on. Their strategy in life is to make loads of spores. They arrive very quickly onto some wood that's just been chopped down. They colonize it quickly, but they're not good fighters. And so when other things come along which are good fighters, they can replace them. So across Kingdom of Fungi, or across the basidiomycete wood decays, there's a whole spectrum of abilities as far as fighting is concerned. And we can actually see these fights. We can study them in the laboratory, and you can see them in the field. So if you saw up, say for example, as you mentioned, a beech tree, you see all these different lines in the wood. And those woods don't just extend across the surface that you're looking at, they extend into the depth of the wood, and they actually surround whole columns of wood. And these lines are, I suppose, a little bit like, we could analogize it to the way in which we humans put fences or walls around our homes, or how we build big walls around our medieval cities. And the fungi are sort of doing that, and they form these things called pseudosclerotial plates, which is mycelium, which is tightly knit and often impregnated with melanin to protect the column of decay inside. And when one decay fungus meets another, if it's the same species, and it's already mated, we won't go into mating in fungi, but let's assume it's already mated, and these two fungi meet, then probably they will just deadlock and just they will stay apart. They are different individuals because fungi are individuals like you and I. But when two fungi which are different species meet, then they will fight because they want to get hold of more territory to feed in. And the outcomes of these battles could simply be deadlock, in which neither fungus gains headway and they maintain their own territory, and they will just decay the volume of wood that they're occupying, and they've got surrounded with their pseudosclerotial plates. But some fungi are better fighters than others, and they will produce, they fight in all sorts of different ways. They can fight at a distance by producing volatile chemicals or chemicals which diffuse through the wood. They can fight at close quarters as well when they actually make contact. Some fungi are parasites on other ones. And the outcome of those battles can be that one fungus replaces another. And so when we look at a piece of wood and see all these lines, you've got to imagine this is what is going on or has gone on to create those patterns. And of course, in the lab, we can set up pitched battles on plates of agar jelly to know who replaces who. Agar jelly is really far removed from the real world, so we don't do that not very often nowadays. We tend to colonize one block of wood with one fungus, another block of wood with a different fungus, put them together and see who beats whom, who takes over the territory. So this is what I like to call fungus wars.
How exciting. And what sort of speed does that take place? Is it something that happens in a matter of days or weeks, or are we talking a year? Can you put it to music and make a Pixar film?
Could absolutely. You can do this in time lapse and for example in agar culture where you can actually see it happening, and I think you can get all sorts of dramatic pictures, movies. So it varies depending on the combinations of the fungi. So there's one fungus called Phanerochaete velutina that I've done a lot of work on. You won't recognize its fruit body. It's just like a thin crust underneath branches on the floor. But this fungus makes these thick mycelial cords, individual hyphae come together, and it makes cords in the woodland. Anyway, Phanerochaete velutina is an amazing fighter, and that beats many other fungi that it encounters. And so, for example, we might take a fungus from an attached branch, such as Vuliminia comedens, and in one block of wood, Phanerochaete in another, and Phanerochaete will just come straight in and just go straight through and completely replace it in a very short time, in a matter of just a few weeks, depending on the size of the wood. But then there are other ones. Something you will be well familiar with, the turkey tail fungus, Trametes vesicolor. If we paired that against Vulamilia, it does beat it eventually, but it doesn't. To start with, the first 2 or 3 or 4 weeks, no headway is made at all in this battle. And then suddenly Trametes seems to be able to, it's got a head of steam. Who knows what? Maybe it's made the appropriate chemicals it needs to, or maybe in some way at the interface of the battle, maybe it's started to wear it down. And then after that time, it then can gradually replaces much more slowly. So they vary considerably in their abilities, and we can make hierarchies of combative abilities of these fungi by doing all these battles. And it's a bit like a sports league, you can think of the football league or whatever sport, and you have your teams at the top of the table who are always winning their football matches or whatever it is, and there's teams at the bottom who occasionally win against some team or other, but not so often. And so you've got the same sort of variation with the fungi. You've got these great fighters and these less good fighters, but those less good fighters actually have other characteristics which allow them to stay in the environment. So they are very tolerant of desiccation, drying out or something. So they have their role, and you will find them there, but they're staying there in other ways. And in the same way as in sports leagues, you sometimes get giant killers. One of these really weak teams normally might come up and kill one of the, well, not kill, would come up and beat one of the really top football teams as a surprise. And you sometimes get that with fungi as well. Sometimes we can make guesses as to why they've suddenly been able to do it. Maybe the conditions are very different and it suits this normally weaker combatant. But it's fascinating. And
it's a bit more than fascinating. So my first thoughts are evolutionary. It's useful that they're not all fantastic fighters, that they have other roles. So that is how things must have evolved, is that some are very good in a very dry environment, some are good in a different environment. They have different attributes, but they're all equally valuable, but some are the real big fighters. The second thing is rather silly. Imagine what you've described to me is pure drama. It's visual, it's got characters, it's got personalities. If anyone listening to this podcast makes films or writes children's books or makes computer games, why isn't there one yet called Fungal Wars? We've had Bugs Life, we've had Ants, we've had many other things. Imagine if this was captured. I know you're a scientist, so forgive me. Imagine if this was captured in a popularist way that had embedded in true science. How engaging that would be for young people, and how might that influence people coming into your field of science and then advance what we need to know. And I think that's hugely exciting. So that's a shout out for anybody listening. This is a field for theatre and drama and art. Wow. Thank you for that.
I love it. And this would be awesome, because I think it's so important that we get people to appreciate how important fungi are. And in some countries, people really are well aware of this, but certainly in Britain, I think that we've got a sort of almost a fear of fungi. We're mycophobes, perhaps, and people often say yuck when I start talking about them, because maybe they think about their food rotting or whatever. But I think it is so important that we realise that without fungi, we would not be here. Fungi are equally as important as plants. We all believe, I'm sure, that we know that plants drive our ecosystems and are important food crops for us. Well, those plants are always dependent on fungi. 90% of them are dependent on fungi. As the Mycorrhizal Association, they're all dependent on fungi for recycling. And I think this is something we've really got to get to grips with, and also to think that we need to conserve and protect fungi in the same sort of way as we do for other organisms. So there's been protection for various plants and animals for a long time, and I understand why people haven't really hooked on to fungi, and I can understand why, for example, pandas, these cuddly-looking animals, really capture people's imaginations, and people want to try and save the panda or save the white rhino or whatever. But we need to save fungi, and in the same sort of way, they are on an equal footing with plants and animals. And actually, a couple of years ago, the IUCN, which stands for the International Union for the Conservation of Nature, took on board what lots of mycologists have been saying, and that is that, we need to conserve fungi. And up until that time, they talked all the time about fauna and flora, but now they feel that it's really important to recognize that it's not just fauna and flora, it's fauna, flora, and funga.
Yes. And in preparation for this, Lynn, I actually looked at the BS 3882 2015, the topsoil specification, the British standard here in the UK. And I read the headline of that, really, and I looked at what we need for good topsoil and how it's defined. And it talked about nutrient the structure, the texture, the organic matter, conductivity, and a test for phytotoxic elements. Now, I didn't read the whole document, but I didn't see anywhere any discussion on mycorrhizae fungi. And this is me, I'm not quite a layman, but I'm definitely not a scientist in this way. But it seems to me that, there definitely is a problem with tree establishment, with newly planted trees, for a myriad of reasons: poor nursery stock, poor planting, poor aftercare, wrong tree, wrong space, wrong soil. But is it also due to some of these imported topsoils, imported to the site, not from country to country, may lack this richness of fungal diversity, so that the newly planted trees only have what they sort of came with, perhaps from a nursery, and they're not able to form these connections, and they may also be isolated from each other, i.e. they may be 10 metres apart, and they're not in a proper community. And I wonder, why aren't we talking about that and an important British standard that is implemented? It's part of the landscape industry.
I think that's a very good point you raised there, Sharon. They do obviously need appropriate mycorrhizal fungi, and indeed the decomposers as well, to do the recycling of the organic matter. And one would think, wouldn't one, that those two aspects really should be a very high priority.
There is a move, which is a big debate. Perhaps we haven't got time in this episode, of introducing mycorrhizae fungi commercially. There is, my own personal view is, it needs to be right mycorrhizae, right place. It's like saying, yes, you've got to have food to be healthy, but you've got to have the right diet to be healthy. Are we introducing something very beneficial, or are we introducing, as I always say, wildebeest in the Antarctica and expect that to survive? Will it do good? Will it do nothing? Will it do harm? I think it's a tremendously interesting area of research. I quite like the use of biochar, which has got that large surface area for soil beneficials to sort of inhabit. But I don't expect you to answer that question. I'm really just having some thoughts with you, really, about this emerging industry, which is commercial, for bringing in mycorrhizae, and also to tell you that in my conversations with the landscape industry as part of my day job, many people haven't heard of it. Now, I think back to star of the show, Ted Green, MBE, who was on recently, and a great friend of us all. And Ted has been talking for a long time about organic plant nurseries, and the fact that if you do have an organically grown tree in its soil, and then you move some of that soil with the tree, it kind of does the job for you. Perhaps that's a way forward. Is the amateur fungal community helpful?
Oh, absolutely. Yes. You would be amazed probably about how useful amateur recorders can be to us scientists. There are very few scientists, but there are loads of people, lay people really, in many ways, who have huge expertise at identifying and a great passion for recording where they find things, when they find it, and on what. And this has really helped us to find out, for example, the effects of climate change on fungi. Now, of course, many people nowadays would say, oh, well, we can find out what fungi are where using molecular approaches. It's no good using fruit bodies, they might say, because fruit bodies only appear now and then. But for the last 100 and indeed more years than that, people have been recording fruit bodies. We haven't had these molecular approaches. And so we can use these records, because that's all we have, that amateurs, used in the sense of people who are not being paid, not in the sense that they don't have skills, because they are hugely skilled. We can use the databases that they have created to drill back and see what communities of fungi we had in the past and what we have now. And it'll be interesting to see what we have in the future. And we have done quite a lot of work on this, both with collaborators in the UK and indeed consortium of people across Europe, where we have looked at some of these large data sets collected by naturalists in the field on fungi, and we found all sorts of really important things. So the very first paper which we published, I had the great pleasure to work with a former professor of entomology at Royal Holloway College, and he phoned me up one day and he said, well, he said, I'm an entomologist. I'm not a fungal ecologist, but I'd really like your help. He said, he was telling me that his father, Ted Gange, was a very keen amateur mycologist, and he'd spent 50 or 60 years, since about 1950, recording with his friends all the fungi that they found in about a 30-mile radius of the city of Salisbury in southern England. And Alan Gange, his son, had a sabbatical and started to analyse his data that he collected. And he said to me, I think I might have found something interesting and perhaps important. Could we chat about it? Would you like to be involved? And I said, oh, yes, that sounds good. And so he came to Cardiff and showed me these data. And I was almost blown away. This was so exciting. He'd found such important things, even just with the first thing that we looked at. He'd done something very simple and looked at, so his father had collected all these data throughout the year, not just in the autumn season, where we typically think about fungi mostly fruiting. And he'd got all these records, and he'd looked at the average first fruiting day for fungi every year since about 1950, and then the average last fruiting day. So they had the first fruiting date and the last fruiting date, and he just plotted this over time. And okay, so the first fruiting day and the last fruiting day jiggle about, because as we know, weather can alter when things fruit, for example. So up until about 1978, it was more or less a straight line, but jiggling up and down for the first fruiting date and the last fruiting date. And then after that, for all the years following, probably up until the present day, he found that the first fruiting date started getting earlier in the year, and the last fruiting date, on average, across all the fungi, was getting later. And so the fruiting season was getting bigger and bigger. In the 1950s, it was probably something like 33 days in southern Britain, and now it's over 70. But that's almost certainly related to climate change. Found other things too. Found that we've always known that some fungi fruit in the spring, but we now know that many other fungi also fruit in the spring as well as in the autumns. So the wood decay fungus, the sulfur-tough fungus, Hyphiloma fasciculare, used only to fruit in the autumn, but it fruits in the spring as well. So what is going on? So people just say to me sometimes, oh, well, that's only fruit bodies. I'm not interested in fruit bodies, but it's telling us something about...
Yeah, it is telling us something.
Hi, I'm Keith Saker, and you're listening to Tree Lady Talks.
Is there going to be an impact on rising CO2? Is that impacting our fungal communities? And what's going to happen then to decomposition? I know it's a massive question, But things that come to my mind, I don't know what sort of questions come to your mind about it.
I'll try and answer a few of those. So you mentioned, is it impacting decomposition? And I think the answer to that is yes. So this business with Hyphiloma fruiting in the spring as well as in the autumn, I think what that's telling us is that if it can make fruit bodies in the spring, that means, I think, that it's been active during the winter, its mycelium, to enough nutrients. So it's doing more decomposition than it used to do. That's what I would imagine is allowing it to fruit in the spring. Okay, so if it's doing more decomposition, and if fungi in general are doing more decomposition because of elevated temperatures, say, that's okay as long as plants are increasing their rate of photosynthesis at the same rate. If they are, then the system will remain balanced. But if decomposers are decomposing more rapidly than plants are photosynthesizing and making new material which falls to the floor, then we are in a bit of a problem, because if in the amount of dead material that's being made, then that means more CO2 is going into the air. So that would be a sort of a nasty, vicious spiral. So that's one thing about decomposition. You also answered, are fungi on the move? And I think the answer to that is yes. Even in Britain, again, Alan Gange and I and some other people have looked at distributions of fungi, and we found that mycorrhizal fungi have changed their distributions, and so have the decomposer fungi, just even on our small island of Great Britain. We know that plants are on the move, trees are on the move up mountains and more north. Of course, their mycorrhizal associates need to move with them, otherwise they wouldn't be able to establish very well. So we know that fungi are on the move too.
Maybe they're up there first and the tree's following.
Well, maybe.
Wow, that's absolutely fascinating. It needs a lot more research. And last year, the most popular Christmas tree decoration was a fly agaric toadstool. And in fact, I'm wearing a silk, beautiful fungi scarf, and it's got into fashion, it's got into home decor. It's seen as being a very beautiful thing, and it's the latest fungi fashion. And I read in the paper this week that there is going to be a fungi film, The Web of Life, being shown first at the IMAX in London, with Björk, the Icelandic pop star, narrating and Merlin Sheldrake presenting. This is all great. I mean, Merlin Sheldrake's book, Entangled Life, made a big impact on me and many people. And it's really great. People are aware of fungi, and it's seen as hot and sexy right now, but we're very fickle people. And I wonder, is it helping funding? Is it helping this essential scientific research? Because it seems to me that fungi are the bedrock, for want of a better word, of life. Are things getting better in academic life for fungi?
Well, I haven't seen any of this extra funding yet, but let's hope that does happen, because as we've pointed out, both you and I, throughout this chat, there's so many areas of research which we really must embark upon, because fungi are so important to us. So I haven't seen any fruits of that yet, but I live in hope. And you are absolutely right that this type of venture, and unfortunately I can't get to see that film, because I'd so like to see the premiere of it. I mean, it's such a great idea. I mean, I think things can only get better. People are beginning to realize the importance of fungi. And we've said the two big reasons for keeping our planet going, but fungi are also important to us humans in other ways that perhaps we don't always realize. So they make important medicines, and the obvious one is penicillin, which comes from penicillium, and countless millions of lives have been saved because of that. But there are other antibiotics made by fungi too, and lots of different medicines as well. And the more we research into it, well, not me personally, but more scientists, mycologists research into it, the more we find in all sorts of different areas. I think there's a fungal product, I think, which is now starting to be used, for example, I think in treatment of Parkinson's disease. stages. But there are lots of different things. There are medicines to stop organ transplant rejection, tissue rejection after following organ transplant. So there are also all sorts of different fungal medicines. They're important foods. We obviously know about eating cultivated mushrooms, but of course there are other fungal food products too. For example, things like Marmite, Vegemite, sort of yeasty foods. That's obviously a fungus. Obviously, brewing, etc., and baking of bread. That's a fungal product using yeast. But there are loads of different foodstuffs. Tempeh, for example, an Asian food. Corn, is a fungal food, a protein food. It's not made of soya. But talking about soy sauce, that's a fungal fermentation. So there are loads of different foods which involve fungi. Many enzymes. Chymosin is made by fungi, and that's what's used to make most commercially produced cheeses. So it's not just the soft cheeses and the blue cheeses which have fungi in them. So there are very many.
Lowe's question of the week.
Lynne, fascinating. I wanted to know, given what you were talking about in just very recently in that chat, do you go foraging?
No, I don't actually.
Would you like to?
No, not particularly. Now, that might sound very funny. So I'm not so much interested in identifying fungi for identification's sake, to put in a name to something. That's not my bag, and I'm not particularly wanting to go out and find things to eat.
And you've just heard Professor Lynne Boddy answering
Lowe's question of the week.
Thanks, Lynne.
And even strange things which you might not think about, but there are, I don't know, some of the chemicals used in paints, and even in some toothpaste. There's so many chemicals made in vast vats by fungi which are used commercially.
I mean, you can make clothes out of fungi, packaging. It's used as building blocks. I've talking about making a house out of fungi. I think it might have been done. And I read in Merlin Sheldrake's book, and I might be wrong, so shout me down if I'm wrong, that fungi can only clear up oil slicks, but even they think perhaps nuclear waste. Well, whether that's right or wrong, the fact is we have the intelligence and some of the knowledge to solve a lot of problems. It often isn't commercially viable. So I hope that the world continues to wake up to the wonder of fungi, for its multitude of uses for us, but also an understanding that everything is a holobiont, every living thing. So honestly, Lynne, I could talk to you for hours. There's still things I haven't asked you, but I mustn't take up too much of your time. But I think I'll end to say, finally, what is your dream scenario?
Oh, that's a great question. I mean, obviously the one would be to have an infinite supply of money to do an infinite amount of research, but that's ridiculous. But I think perhaps more important than that is something which we've just been talking about, really, and that is that everybody realizes the importance of fungi, and that we actually start to work with fungi in mind to better our environment. Of course, we've steered well clear of some of the negative impacts. We agree that there are negative impacts of fungi, like some of the crop diseases and animal diseases as well. But taken on balance, we will not be here without fungi. We want everybody to realise this and to take this on board and to consider fungi in the same sort of way as that we would with plants and animals.
Professor Lynne Boddy, it's been an absolute privilege to talk to you. Absolutely fascinating. Thank you so much for your time.
It's been my absolute pleasure. Thank you.
A few weeks ago, we interviewed Woodland Heritage, and one of their team is on a big fundraiser for that charity. So Charlie Dedman, he's a furniture designer for Gaze Burville, and he's walking 630 miles along the South Coast Path to support woodland heritage from the 1st of April. And you can give at justgiving.com. Just put in Charles Dedman, that's Dedman. Good luck, Charlie.
All right, Charlie.
It's a great cause.
And just before we go, time to tell you about our very exciting new conference in February 2024.
Trees, planning and construction. Is it really working? Do you feel isolated? Well, this conference plans to change that. We're here at BS 5837 Speed Briefing. Biodiversity net gain, what does it mean for trees? And trees and planning law, are you up to date? Sarah Dodd will tell us. Hear from keynote speaker Keith Saker and Jeremy Barrell. We'll hear the views of London Tree Officer John Ryan on what works well and ideas for the future, and a panel of experts, a planning consultant, highway engineer, demolition contractor and building contractor, and I will be saying what works for me. All this and outdoor demonstrations on the 20th of February 2024 here at Braxted Park. And thanks to our headline sponsors, Bartram Trees, we're offering cut price tickets for tree officers. See us at Sharon Hosegood Associates /training or Eventbrite Trees, Planning and Construction.